I've always found that the percent saturation is kind of a misleading relative term. I prefer to work in concentration units. So hypoxia or oxygen minimum zone is what, less than about 20 to 25 micromolar. And we've been working in pretty well oxygenated water since then.
Were you in an oxygen minimum zone earlier on this cruise? Is that what you're referring to, or?
Yeah, we've been, we've been working in oxygen minimum zones pretty much all the way down the coast, from as far north as the Olympic coast all the way down here. We've been working in pretty low oxygenated waters in the 300 to 1500 meter depth range broadly.
Yeah. For people who don't know what that is, can you explain that? Like why are these areas of low oxygen found in the deeper sea?
Yeah, definitely. So in certain parts, usually along eastern margins of oceans, where there's usually enhanced upwelling due to oceanographic circulation patterns, wind-driven upwelling, it can create high productivity areas, and that productivity, when it sinks down, is degraded by microbes, which draw down oxygen in the water column. That drawdown of oxygen creates low oxygen areas that vary in depth, but typically most strongly between 3 and 900 meters seems to be the trend. And in certain parts of the world, if there's upwelling of rather poorly oxygenated waters, combined with that drawdown of microbial metabolism, drawing down oxygen from the surface and upper bathyal depths, it can lead to zones of persistent low oxygen, which are called oxygen minimum zones. And I think, they're defined, how you define them is slightly different depending on what units you're looking at or where you are, but typically I think less than 2 milligrams per liter is an oxygen minimum zone boundary, and like I said, 20 micromolar, 20 or 22 micromolar also has been identified as a boundary of the oxygen minimum zone. And the major characteristic between these oxygen minimum zones and just zones of low oxygen worldwide, it's pretty characteristic to have a drop in oxygen as you go deeper, but the oxygen minimum zones are persistent through time. And so you have depths that are persistently at hypoxic or even sometimes anoxic levels through several months out of the year, through, maybe even just persistently through time. I'm very curious about the persistence of some of these oxygen minimum zones if they change seasonally with changing winds and circulation patterns. That's been suggested, but a lot of the times we need more time series data to better understand how variable oxygen concentrations are within the minimum zones.
It's really, it's really interesting, and how animals, the midwater organisms adapt, and whether they're in or out of the oxygen minimum zone, how long they can spend there.
Surprisingly, we were actually observing quite a number of deep water corals, Paragorgia species up in the Olympic coast, associated with some pretty low oxygen concentrations, 4 to 5 micromolar, and notable changes in community structure as you increase or decrease oxygen concentrations over the depth gradient. And yeah, I mean, it's very interesting. It's definitely becoming more of a concern, I think, at least in the coral community, trying to understand how coral communities change across the oxygen gradients, because certain parts of the world are experiencing deoxygenation of surface waters and deep sea waters, whereby as warming waters hold less gases, so it reduces oxygen concentrations in those waters, and changing circulation patterns can actually reduce the oxygen supply to certain parts of the water column and certain parts of the world.
Yeah, that's super interesting, especially as we expect more areas to become deoxygenated with the warmer waters. Lizzie, are we going to start a transect?
Yeah, I've been watching the navigation screen, and I think how much longer? Probably until we hit roughly that target, that little bump.
We've got about 230 meters. So
Oh, perfect.
I think we want to do one more bump south, 30 meters, just so we get in the right.
Keep in mind, we'll probably swing like 20, 30 meters.
Yeah, that's fair. So maybe we can just start that move east now. Southeast to the target. What do you think,
that
Bob?
sounds great. And
Or do you think we need another step? Swinging 20, 30 meters is accurate at this depth.
Sure. Yeah, I think. I don't know.
Let's do a short ship step. So this.
Just say, what this percentage of the depth, right? 28. We're at 28. 2,800, 380, 3,800. What is that? 2,800 divided by 3,800 times. What did we do before? 40.
What's the 40?
The meters. If we say we're not going to swing as much here, I don't know. You're trying to get a number out of me.
Let's do another short ship step down. I erased it.
Oh.
Let's do another short step down, just so we're not overshooting, or we're not undershooting.
Okay,
Bridge nav.
that sounds good.
Two 0 meters, 180. And then once we finish that move, we'll have about 220 meters, so that'll be a perfect time to run transect to that target on the orange.
Cool.
Up there.
Sounds great.
To get a number out of Bob, you're gonna have to pass his exam on the hydraulic systems of Hercules.
Clearly not there yet.
At least we're seeing some rocks now. Out of that bright blue zone, and now we're getting into like a yellowy, slightly higher sloping area. Seeing some anemones, brittle stars, that, oh gosh, I'm gonna have to go back and look at my notes. Radicipes, is that what it's called? Yes, radicipes. Sea pen?
It's a chrysogorgid.
Oh, a chrysogorgid.
One of the golden corals, they're called.
Oh, it's a golden coral.
Yeah,
Awesome.
it's kind of a confusing, there's actually many different groups of corals. Soft corals are called gold or golden corals. But yeah, golden corals are the chrysogorgids. And
they're not soft.
They are soft corals, but the axis is typically has a metallic luster to it. And so if you remove the tissue, it has, fairly metallic color luster.
Okay. Thanks for that.
Not to be confused with the gold corals, which are the corals that you typically find in the Pacific Islands and off Hawaii, which are actually zoanthids that form. They parasitize bamboo corals and then form and precipitate their own skeleton onto the remains of bamboo corals and continue to grow.
Oh, that's fascinating.
One of them, yeah, there was a genus that was described or established in the past few years called Kulamanamana, which is the Hawaiian gold coral that we're now finding is actually quite extensive throughout the Pacific Islands.
So it's not really a coral, it's a zoanthid. It just deposits a skeleton on top of the bamboos.
Yeah, they seem to parasitize mostly bamboos, which is really interesting, that specific association. There are other types of zoanthids, of course, that parasitize all kinds of corals, paragorgiids, as well as corallium and coralliids. But this one, Kulamanamana species is usually found on bamboo corals.
We'll see some gold zoanthids on the gorgonians and sea fans here in the Channel Islands.
Are we on a
You see them
transect?
on. Not yet, right?
We have not sampled any zoanthids that I've seen. We saw some anemones that looked zoanthid-like, but we never collected any, at least 3 or 4 dives ago. I'm not sure exactly where. But no, I haven't seen very many zoanthids, which has been interesting. Maybe it's a depth thing.
Yeah, it could be. We definitely see them in the shallower areas where there's high densities of sea fans. I did not see any in the deeper dives on any of the dead corals, so maybe you're right. Maybe it could be a depth limitation.
Yeah, I think some of the golden corals off Hawaii have actually been aged at close to 2,000 years old. I think.
Oh, wow.
I have to check my data on that. But there's some of the more longer-lived Cnidarian corals that produce this skeletal material.
Black corals can get pretty old too.
Yep. Yeah, black corals and these gold corals are some of the more longer-lived, but it's difficult because oftentimes to get good ages on some of the oldest colonies, you have to sample some really old and large and long-lived stuff that's not exactly environmentally friendly.
No, and it's for aging, you really need the base, right? The base of the coral.
Yeah, for most things.
Most things, yeah. So there's really no way you can't clip an arm necessarily to figure out the age. You have to sacrifice pretty much the whole organism.
Yeah, bamboo corals, definitely. Black corals, yes. It's difficult to tell, ages on most things.
Yeah, I can imagine.
I think fisheries bycatch has actually been successful at providing a lot of the material used to age a lot of the older bamboo corals and older, larger colonies that get caught up in trawls and things like that.
Yeah. It's good to know that they did go to a use, though. If they had to come up, we did learn something from them. Looks like there might be something big coming up to the right. Big-ish. Yeah, and we're not on TrackSec right now, and so we can zoom and explore until the ship move is in place and we're ready to start.
Yeah, we've got about 10 minutes to settle before we're able to start that transect to the east.
That sounds great.
Maybe a little less. Then I think that'll actually be perfect timing to complete that transect before the next watch. That should be doable. Should be about.
Great.
Cheetos.
They put up more Cheetos?
Sure did. How many are there? We need to leave
Cheetos.
now.
Oh, are these those tunicates?
Oh, there's another horsehead tunicate.
Two
Two of them.
Yeah.
Wow.
Oh,
Sam,
yeah,
most
we're
of the chips are
surrounded.
in his
Early.
cabin now.
Up. Science, we want to zoom on one or both of them?
Yeah, why not?
Bob, did you take all the chips? Yeah, I knew you did. I had to read your lips to hear that.
Sorry.
It's okay.
I said I took them all and stashed them. You get nothing.
I knew it. I knew it. No respect for the videos, huh?
Okay. Raj. Where am I getting yanked about? Oh, we're way out in front. Yeah, I didn't check out the weather very good when I was out there. What are we doing?
Still looking like we're bouncing between 18 and 20 for wind.
Good sailing winds.
Yeah? That's your numbers?
Depends. If the seas are relatively flat, that's great. The boat will take off like a rocket.
Want to zoom in on that thing? So is the intake on the left there, and this is the exhaust, or how does this thing work?
I'm wondering if it's actually just a carnivorous species. That they, maybe the organisms go in and they don't come out.
Yeah, because I had that same question. If this is like facing the opposite direction of the current, how is it filter feeding if there's not another intake area on the other side?
Yeah, it seems like because
a
we're
caution
always looking at the same side here, and we're going into the wind.
Mm-hmm.
You think it's open on the other side? We need to look at it from that side?
We could.
Can you zoom out?
If we've got time before that
great.
final ship move.
It's kind of an odd-shaped rock that it's on, too. Semicircular.
Are we feeling about?
Yeah, it looks like a...
Can I put on that ship move, pilots?
Sure.
Yeah.
I'll just quick look over here.
Does it feel like it's getting warmer in here to
all
anyone?
secrets.
Yes, but I'm
warmer.
watching the racks and they're cooling, which kind of doesn't make sense.
Are you all just getting excited by the anticipation of the next hotspot for the slope we're encountering? Just me?
I'm optimistic.
It's very shiny, this tunicate.
I think it was Erica. I'm trying to find the place in the chat. It kind of looks like Snoopy.
This thing turning
Snoopy's
like
head on
so
the
I
side.
can't get a look at the other side. Like, nope, you're not gonna look in there.
It's special.
It'd be great if we could catch it in the act of feeding.
It's got a hole on that side too.
Yeah, I bet that's the intake and
there it is.
There's the exhaust.
Oh, wow. Okay, so it does have one.
Yeah.
Looks like a little balloon animal.
We zoom in now.
Look at that. That's awesome. Like a Macy's festival gone wrong. Macy's Day Parade. There we go. No, not Macy's Day Parade. What am I saying?
Yeah, that's the one. From New York,
Oh,
Thanksgiving.
yeah.
Yeah.
Macy's Thanksgiving Day Parade?
Yeah.
Because there's no Macy's Day Parade.
We're affecting the current here.
I'm going
Ship move has started.
Unfortunately, they had incidents like that on that parade too.
Yeah, when the wind picks up.
And what's the
Okay,
new
gotta
bearing,
move.
Sam?
101, please.
101.
Thanks for that. That was informative. Yeah, so I can see why Lisa picked this habitat. It looks like there's a lot of hard habitat, just not very deep under the sediment when this isn't a hugely significant slope. So I feel like I bet there were hard bottom returns here, but it's just also a very sedimented area.
Are we going to start a transect here?
We are getting lined up. We got lasers on. When we start moving, I will drop a target. And it will be transect
9.
9.
9.
The AC just kicked on.
When we get going, can we zoom in a tiny bit?
Zoom in, Al.
Just a little bit.
Stand by for Al.
He's over here.
Oh yeah, no worries. When just before we start transect, that's all.
Lasers. Oh, I think it was just a junk zoom or a full. Yeah.
I think we're zooming in on this thing.
Oh, never mind.
I think.
Yeah, it's okay. We haven't started the transect yet. We're going to wait until the Argus starts moving again.
Yeah, we've
Is this
got
what
time.
we were looking for?
Oh, it's just a bit of seagrass and some
Tight.
urchins, some very tiny urchins.
Yes, that's all the way in.
Or
I don't know what we're
small
looking at.
box.
Zoom out.
Coming out.
Yeah, I'm good. Thank you.
Junk zoom.
Don't zoom out.
So we're in the last hour of our watch shift. Laura Francis and I were both with the Channel Islands National Marine Sanctuary. And we'll have some other folks coming on in about an hour.
What was our bearing?
101.
Oh, I did it again, didn't it, Paul?
101. 101's this way. 101. 102. 101. Close enough.
That was the only time this watch, right?
No, it happened just like 5-10 minutes ago as well.
What are you talking?
Twice this watch? Okay, thank you.
What are we talking
The
about?
top monitor is like cycled on and off twice.
Do you think it was power or just video went away and came back?
Well, the video didn't go away on the bottom screen or freeze or anything, so.
Different route though. Okay. Yeah, I saw it stayed up over here too on the quad.
Are we moving, Bob? Transect is starting.
Wonderful. Thank you.
And lasers are on.
Lasers on.
Perfect.
So now we're in transect, right?
Now in
Yeah.
transect.
Yeah, now that we're on transect, how many of us are going to have to go judge the weather from the mess porthole?
Those of us that don't have Cheetos.
Oh, is the weather fine? But that's the cover for the Cheeto run.
Yeah.
Oh, I see. Because we know that the weather's going to pick up. Because we're still waiting on the 12:30 call today, I think, to make the judgment as to if we're going to do another dive in the Santa Lucia area or head straight to Channel Islands. So I was just like, wow, they're really watching that wind for anticipation of this 12:30 call.
Well, we are, but,
Which I
you know.
see. Oh, but I mean, yes, I see.
Yeah.
Yeah.
We've actually been tracking the weather over the last, well, since the dive started. Every 45 minutes to an hour, the nav has been taking notes on it. And yeah, we're still, it definitely got worse overnight, but it's dropped back down.
Hmm.
It looks so calm and peaceful down here on the bottom.
Yeah, there's another significant weather system moving in coming from the northwest. To the point, yeah, if we once you guys do arrive to Channel Islands, we will probably be limited to the eastern part of the sanctuary again. Not as bad as the last time the Nautilus had to run to Channel Islands, but Santa Rosa might be workable, and then east versus Santa Cruz and east. So we'll be making some calls. You can see some brittle stars kind of half buried in this sediment. We're still coming up on some ledges, which is more interesting than what we were seeing before. Nice grenadier coming into the mid-screen now. And we're doing a quantitative transect, which includes, or the protocol is fixed height above the seafloor, lasers engaged, and then we're trying to hit a distance of 200 meters and as roughly as straight as we can make it. So we have a estimate of how much ground we've covered to be able to then estimate if there are any corals and sponges in this area and identify the habitat that they're on. And speaking of transects, if anyone is interested or a formal or informal educator, Laura, we have some transects and some species ID guides on our website if anyone was interested in practicing or following along with some of the transects that we've done in the past.
Yeah, we have some, if you go to the sanctuaries.noaa.gov website and then look under education, you should be able to find a deep sea coral communities curriculum guide that has a whole lesson plan and PowerPoint and lots of information and background materials for teachers. And then, as Lizzie mentioned, there are some underwater transects from all of our West Coast National Marine Sanctuaries that we've dropped off for a second there.
There you are.
That we've pre-recorded some video transects, and you can watch those and do what the scientists do, count and measure the species along the way on the transects. And then you can also compare between the different sanctuaries, the habitat types, and also the different species that you might see from north to south, from Olympic Coast National Marine Sanctuary off the coast of Washington all the way down to Channel Islands National Marine Sanctuary. So some great resources there, including field guides. And Lizzie and I are working on a cool project creating some 3D printed corals. And so, Lizzie, do you want to talk about the process for making those models that we're working on?
Sure. So in addition to this awesome curriculum that Laura was just discussing, we've been thinking about ways to bring this really remote system to the hands of the public in an engaging and interactive way, because it's not really a habitat many can relate to unless there's a lot of people, which I'm sure there are, that really enjoy the live broadcasting from ships like the Nautilus or maybe the Okeanos. But for a lot of others, this isn't an easily accessible kind of habitat to think about or potentially care about. And so we are going to make 3D prints of some of these corals, potentially sponges. We haven't quite figured that out yet. And I was actually contacting where the Nautilus archives a lot of their samples, MCZ with Harvard, and we'll have the possibility of getting some loaned to us to make these 3D prints, which starts out, we have a partnership with someone on the UC Santa Barbara campus. It's called the Innovation Workshop. It's in a microfluidics laboratory where they do a lot of micro printing. They create channels for fluid dynamics for a number of different engineering and medical applications, and they have a ton of different 3D printers. And they're branching out and using these tools for a lot of different applications outside of engineering and medical applications. So for example, I'm going to be partnering with them to scan and print coral skeletons or preserved specimens with tissue on them. So far we've used the skeletons of Lophelia, some samples of this hard coral that's pretty ubiquitous on the West Coast and in the Gulf of Mexico. And they have a really nice setup where they have a software and a tool that has a camera on it, and then it's attached to this pedestal that you place your object on, and then you set the software on a computer that's attached to how many degrees you want for each rotation. So one rotation would be 360, but we kind of divide it up into smaller pieces of the pie where we take multiple pictures where parts of the object are overlapping, and the computer program stitches them all together. And so the more rotations, the better idea it has of where things go. But I think even 10 rotations, gives me a pretty darn good model. And to even get the top and the bottom, you just flip the object over or put it on its side, and it recognizes, based on the points it's collecting with this camera, what part of the pictures belong to what, and it just auto stitches itself together into what we call a watertight digital image model, which means it's completely closed. There's no holes, there's no gaps, which then allows us to take that and send it to the 3D printer, which it will then figure out the best and most efficient way to build or print this coral that uses the least amount of travel time of the resin needle, the head that melts and disperses the plastic. and uses the least amount of plastic at the same time. So it builds it up little layer by little layer. And we've done this with Lophelia, and it's turned out really well. And so we're also interested in collecting or borrowing other specimens in archive to create more 3D models. Because we do have a few at the sanctuary, like Lophelia, except those are pretty darn fragile, and we only really have one set of them, but they're highly requested from the sanctuaries across the West Coast. And so we're thinking about, and in the process of, creating these educational kits with several species of 3D printed specimens. And so we're hoping to bring these models into classrooms and public outreach events so people can really get an idea of the shape, the polyps, the structure, the branching of a couple different species to help them better understand this environment and understand the functions they perform within this ecosystem. And I think
I'm
we
really
have
looking
other.
forward to getting those. Yeah.
I know, me too. And and then COVID and work from home, so the lab was shut down. It's just now starting to open back up. So we have one other scanned species, which is the Desmophyllum dianthus coral, which is the coxcomb cup coral. We have a couple of those, and Lophelia, and so we're still looking to find roughly a small, maybe the size of my hand, the palm of my hand and my fingers, size of like a bubblegum coral, I think would be a really great example, since we can find those everywhere, because we have to balance the right shape. Too much branching would actually be too big of a file to print. It's too complicated. And so this example of this coral on the left in the middle of the screen here may not be a great example. The branching is too fine, but I think something similar to that, or maybe smaller than that, could potentially be a good candidate. And so we might scan them to create the files just to see, but then it's really the printer that has to upload and digest those files to then create the directions for the actual printing itself. That's kind of where the limitation comes in. So we're happy to create those files, and we'll probably try to borrow some of these specimens to do that, because then we can also share the 3D images online, which could be useful for a number of different applications, morphology. It's just also a really cool way to see and interact with these corals, even if you don't have the physical model in hand. And so we'll try to make a subset that we can print, but we'll be publicly sharing all of the digital images that we make, so they're available to anyone who's interested.
I know some schools have 3D printers now, so it'll be fun for them to get a hold of the files and be able to print them themselves.
Yes, that too. Like a lot of those Ultimakers 3D printers are available in a lot of science classrooms. So that would also be an awesome application for the schools that do have those resources.
And we'd love to host those too on the Nautilus Live website and our education resources and our expedition page to link back to content that's created. So keep us in the loop. Sounds like a really exciting project.
Yeah, definitely. Just gotta get back in the lab. So
yep.
I think what they're doing now is they've hired, because this lab is really awesome. It's mainly meant for engineers, but they have what do they call them? Workshop wizards, which is a team of undergraduates that get paid by the hour to do some of these projects. Since we're not allowed to step in because of COVID, it just keeps them safer and reduces their exposure to more people. And so we'd be happy to support them and get them engaged and start scanning and printing some of these things. So I think we'll be moving forward shortly with this project. And I'm trying to think, I wonder if you have any suggestions with since you all interact with a lot of the collections that come up, are there any glass sponges that aren't too delicate that they just kind of shatter when you collect them, but kind of hold their shape when they get to the surface? Because I know a lot of demo sponges will shrink and shrivel, and they're not really recognizable as they are in situ. So I'm wondering if you have any examples of potential glass sponge collections that we might be able, that would be rigid enough that we could print, scan and print.
Yeah, yesterday was a really incredible dive for sponges. We had a nice Faria that came up pretty whole. It was about 5 centimeters across, 5 centimeters wide, and it fit perfectly in a jar, with a lid. So small enough that you might be able to print it, but complex enough that it has some interesting structure to it. There were a number of small ophiuroids inside as well. We didn't attempt to take them out for a chance of breaking it, but for the most part, it was pretty intact. I can get you a sample number on that. Although it was the only Faria that was collected yesterday on 1831. So I'll be posting the dive log, sample log, probably sometime later today up on the Google Drive, and you can just take a look at that sample number there.
Okay, yeah, that sounds great. I'll write myself a note. Yeah, and if it was collected for purposes, for genetics, or it needs to go to someone else, I totally understand. But maybe we can, was it collected on a rock? Is that how it came up so easily?
It actually was cradled gingerly by the manipulator, so much so that it was actually really well preserved. Wow. It is in ethanol, right? But yeah, maybe you can
that'd be fine.
Coordinate with Abby about that.
Yeah, I was talking to, I believe his name is Adam, with where Nautilus archives the MCZ, and he said I would have to get him specific specimen IDs, looking through their archive, their database. But he said briefly taking him out of the ethanol for like probably max of 10 minutes to get the scanning done shouldn't be an issue.
No, yeah, I think that'll be okay.
So, yeah.
Yeah, I mean, as far as I know, the only people who are requesting sponges are you and Abby, and I think that would be okay.
Okay, yeah, that sounds great.
One thing I've always wondered is with these really fragile glass sponges, or even sponges generally, how much of the material do you actually need to do a good DNA extraction? Because there's a lot of spicules and things. I wonder actually how much of the mass of a sponge is DNA. Do you need a lot, or is just like a small 25 milligrams or something sufficient?
Got it. Yeah, I also wondered the same thing. I wonder if Meredith is listening and she can chime in. Meredith is saying not a lot. She's been surprisingly successful at getting DNA out of glass sponges.
Yep. Great. Yeah, we've tried to, we're making sure that every single collection has a voucher for DNA and ethanol, and then a lot of the large sponges that are just too big to fit in a jar, we've been drying out, and we've actually had really good luck with this one extremely large Faria we collected at Davidson. We dried it out, and actually it has it's not as fragile as I thought once they're dried. The skeleton is fused, so they have some structure to them, but that one's going to have to go in a box all by itself because I'm concerned that it might get crushed if it's in there with anything else for shipping.
Right. Yeah, because some of those heterocones, like the really small juveniles that look essentially exactly like the adults, but very small, I figured those would be great because you don't lose too much given that they're small, so it'd be easy to print. But Meredith was saying that those get very easily crushed.
Oh yeah, the heterocone is not good. Most of the heterocones that have come up in just like piles of fragments.
Yeah.
So we do our best to put them in jars to keep them whole, but
Yeah, so if we see any smallish sponges that we think, Steve, that you think would be a good candidate for that, we can go ahead and collect them, and that they can get archived with MCC, and then I can request them after the
Yep.
fact.
Yeah, all of, so when you get the data drives, all of the lab photos will be available, so you can take a look exactly how it was when we got it up on the surface, and you can also make your decisions that way,
Oh,
because some
yeah.
things look very different on the surface versus on the seafloor,
Yes.
even if they're structurally intact. The color drains, or they're deflated.
Yeah, yeah, the deflation would be the biggest issue. So that's actually a really good point to familiarize myself with what they look like when they come up, so I know what to request. So yeah, and the color isn't as big of a deal. It's because we have a limited number of colors to choose from in terms of resin. So we're still thinking about if we want to paint them to make them more realistic looking, or how we might want to do that. But the first step is just getting them printed.
Well, and keep in mind too, for large objects underwater, a sponge might be hard because there's a lot of kind of internal features, but with like a coral, just being able to like take a lot of images from different angles around it underwater, you might be able to then still get that 3D shape without having to actually remove the sample too, or for larger objects. So
Yes. So my experience is more limited. I know very well how to use their specific scanner, which plugs directly into their printer. But I know there are ways, if you can do a little bit more of like post-processing, once they do create like a structure from motion model.
Yeah, exactly.
So long as the resolution is good enough, it could be interesting to print. But we'd have to make it watertight, which I'm not sure what programs I would need to do that. It'd definitely be worth looking into. But that would take some more effort, I think.
Yeah. I think the hard part there is just getting enough angles and pictures that the structure for motion is going to come out with a good result. But once you have the structure for motion file, you should still be able to print it. Then the geometry of whatever you're trying to print how good the print comes out will depend on the geometry of the object itself. But
yeah, I forget how many million vertices there were on Lophelia, because it does have empty space. It's a lot of polyps kind of chaotically strewn about, at least the sample that we have. And it did have to reduce the resolution. The original looks amazing, which I have those files I can share with Nautilus. But to print it, we definitely had to decrease the resolution to make it a manageable file size for the printer. So the scanning is able to do amazing things, but yeah, we're more limited with what the printer can do.
do you know which printer they're using?
I have the paperwork because they have 3 different printers, and I've done the trainings to use them, but it's been, how long have we been in quarantine? I think the last time I did a training was in March. So I just haven't thought about it since, unfortunately. So no, I don't know off the top of my head, but they have the really interesting, like multiple different printers where you have like the resin that's, you string in the like a thread of resin almost to the head that melts it, and they print it.
Yeah.
They have ones that are almost like hanging upside down. You have the base, and it's sitting in a pool of resin, and a laser essentially draws on that layer, and the laser is what is polymerizing the resin onto that base, and then it lifts up millimeter by millimeter, and it's just coming out of this pool of resin. They have multiple different.
Yeah.
The whole thing just blew my mind. It was so cool. And I had no idea that was a resource here at UCSB. And then they're happy to work with any applications. someone was looking at microplastics in terms of how, like the wear and tear of the foam, the synthetic foam soles of the outer soles of the shoes. And so they were taking brand new shoes, creating these 3D scans of the shoes, and then they were walking around on it for certain periods of time, and they were taking the scans again, and they were calculating the volume changes where the shoe was getting worn to estimate how shoe soles were contributing to microplastics in the environment.
What are the white fan-shaped, are those corals that we're seeing? We passed a few of them.
I think so. Those look like the pronoid corals. Steve, in his observations, is saying the Narella sea fans. Have we collected a yellow-looking bathypathes? It might be heteropathes.
We did in box
Okay.
starboard A. We collected one
Okay.
in the first watch.
Okay, cool. Maybe if we see another of those droopy yellow bathypathy looking corals, we could sample when we're not on transect.
Yep. Yeah, that one looked a little ragged. I wasn't sure if it was either just a very small colony with really dense branches or if it was something else going on there.
Mm-hmm. And Nicole was in the science chat, the expedition lead, and she was saying it looks like we are going to transit to the other location on top of Santa Lucia Bank for the next dive. We were waiting to see if what the weather was going to do, but it looks like it's at least worth transiting there to see if the weather holds for a potential 8-hour dive on top of a suspected petrale sole area, mating, breeding area.
How deep is that dive? You know?
I think it's in the 500-meter range. It's pretty shallow, closer to shore and much shallower. Yeah. Yeah, Lisa's confirming 500 meters. So that's what you can expect with the next dive, which I believe, is it 4 p.m.? No. 8 p.m.? Yes. 8 p.m. to 4 a.m. It's an 8-hour dive. It's on the shorter side, Lisa's saying, but it'll also be shallower, so not so much time will be taken up by ascent and descent. Lisa's confirming they aren't sure about the weather coming in, but I think, like Nicole said, it's probably worth transiting there and assessing once they're on site. We'll learn more every day at 12:30. Nicole holds a dive briefing to talk about a debriefing for the previous dive or the dive that we're still on, and then planning for the next dive that's coming up after it. We'll talk about weather, waypoints, any questions we might have, any complications we think we might come across, like fishing gear, that kind of thing. So that we'll be learning more at 12:30 today.
So our transect is complete. And we were kind of doing a little test of our USBL nav system here to see what the offset was. So we've actually moved over that kind of orange target we had originally identified. And either there was no slope there to speak of, or our offset is less than we thought it was. So it was a good test, but inconclusive. And now we've got 20 minutes left. So would you like to start heading down to this other waypoint that we had dropped, another orange hotspot?
Sure. So we have 20 minutes left in this watch, right?
Mm-hmm.
But really probably like an hour 20 left in the whole dive.
That's correct if we're going to start moving now.
Okay. Could we make it that far to the second orange spot within that time frame?
Let me do a quick calculation. It's 470 meters.
Oh, okay. So we're closer than I thought we were. Sure. Why don't we try to end there?
Roger. I'll put in a move and then I'll get an estimate on time here.
Okay.
Bridge nav. Five 0 meters, 135. .135 is our new heading. Bearing. It can be your heading if you so choose.
Can you explain the difference, Sam, with the heading and bearing?
Sure, it's something that I have recently learned. Heading is the direction, in this case, that, we'll use the example of the ship. Heading is the direction the bow of the ship is pointed in, and bearing is the direction it's going. So in this case right now, the ship's heading is northwest, and our bearing is southeast.
Oh, I see. So the ship's kind of going backwards.
Ship's going backwards, but our heading is still, yeah, northeast.
Got it. Got it. Okay, that's really informative.
Yeah. Hasn't sunk in yet for me, though, so sometimes we'll tell the bridge heading, and bridge asks, are you sure? Are you sure you don't want bearing? So,
Yeah.
Always learning.
Because usually the bridge has control over the heading of the ship.
Yes.
They're dealing with the weather.
Can we take a look at 9 o'clock, this white stock?
Can you zoom in now?
So is that a bamboo coral?
Yep, that looks like a bamboo coral.
You can see down on the base here.
Yeah, there's a small,
A little tiny rock and a tiny black coral.
tiny black coral. Yep.
That's awesome.
And your friend, Mr. Sea Spider.
Ugh.
Hello. Hello,
Do
friend.
we have a
These-
few different kinds of bamboo coral here, or have we seen several different types on these dives?